I just wanted to give a few details about the Skaggs Fellowships and the significance that it has in the college, and also just a little bit of information about how prestigious and competitive these awards really are. This endowment provides four full fellowships per each year. So each student gets essentially full stipend support for 1 year. So for a year for 4 students, that's really an impressive amount of money. And the endowment that it takes to kind of support that level of payback is really significant. The fellowships are quite prestigious, and there's very strong competition amongst the students for these fellowships. The recipients of the fellowships should be very proud to have been selected for this honor. The selection committee each year has a challenge to identify the 4 recipients because of the quality of the applicants is so high. And so it's not the situation where we get, 2 or 3 students who are really outstanding, and then, they get the awards, and, we just kind of dole them out to, kind of whoever fills out an application. The quality of the applications is just outstanding. And each year we have 10 or 12 applicants that are just really excellent. So we really do struggle amongst such high quality to try to identify 4 people that kind of rise above and that deserve this recognition. And also the criteria are pretty strict. They state that the students have to have passed their preliminary exams in order to be considered. But additionally, they also have to have at least 1 year left to finish their dissertation work. So students typically complete their prelim exams in year 2, and then they still have to have at least a year left. And so for each student, they usually only have 1 or 2 years that they're really qualified to be competitive. And so that really narrows the pool. So the fact that we each year have 10 or 12 students that fit these criteria that are really outstanding is quite impressive. So the students that are successful, again, should be very proud because the competition is very stiff. And so finally, I just want to congratulate the 4 recipients for this year, and I'm sure that we'll all be very impressed by the presentation of their research projects.
Great, thank you very much, Daryl. Well, let's jump right into the presentations now. We'll, I think, first hear an introduction by Dr. Kristen Keefe.
So thank you, Randy. Thank you, Ronnie, and the AllSam Foundation for supporting our students. I certainly appreciate it. It's my pleasure to introduce Danielle Giangrasso, who's a neuroscience program PhD student working in my laboratory, who's going to start us off. Danielle got her undergraduate degree summa cum laude from Appalachian State University in psychology, and really has moved herself into kind of harder science in the neuroscience program here, and I think has maintained a 4.0 GPA throughout her neuroscience program here. In addition to the Skaggs Research Fellowship, she was a recipient of a Roy Kuramoto Award from the college. She also was a recipient of a slot on the National Institutes of Health T32 through the neuroscience program, training program in neuroscience. And as an undergraduate, she had a National Science Foundation research experience scholarship that let her go from her home institution, which isn't so research intensive, to the Medical University of South Carolina to start building her research experience. So throughout her academic career, Danielle has really shown a commitment to doing research. Since joining the graduate program here, she has middle authorship on 1 paper, first authorship from the initial work that she did in my laboratory, which was published recently in the Neurobiology of Disease. She has a review article in preparation that I'm anxious to see. I can't wait to get my hands on it, and has made a number of presentations at national and international meetings. She also, in addition to her research, has completed the Higher Education Teaching Specialist designation through the Center for Teaching and Learning Excellence, and also has completed a year as a Science Communication Fellow through the Natural History Museum of Utah, because she has a really intense interest in bringing science and making science accessible to the public. And I think we'll see her in her future career path as she finishes up her dissertation work and moves on, being a big voice in bringing science to the public. And so without further ado, I'll turn it over to Danielle, who's going to talk about, a lot of new stuff that she's brought to the laboratory and in a collaborative project between me and Karen Wilcox's group. Oh, I should also note, we learned yesterday that she is also known as Prince Verma. It came out on Facebook yesterday, so it must be true. And we're waiting to hear kind of what throne she might ascend to. And we're hoping she'll make big contributions financially then to the college as well. So hopefully she won't be sending any bank account numbers to the people who set up that Facebook account. So with that, Danielle or Prince Verma, I will turn it over to you.
Thanks so much, Kristen. My Facebook account was hacked, so no one except Prince Verma, their friend request. But let me go ahead and share my screen here. Okay. All right, here we go. So hello, everyone, and thanks for coming. And thanks again, Kristen, for that great introduction. I wish you could walk around with me always and introduce me like that. That would be great. So as she said, my name is Danielle Jean Grasso, and my project is focused on glutamate dynamics and behavioral control in cocaine abuse and addiction. But before we get into any of that, I was wondering, have you ever driven home from work without thinking about it, as if you were on autopilot? What about when you walk into your bedroom? Do you automatically turn on the light without needing to see where the light switch is? So these are examples of automatic behaviors, behaviors that we've done so many times that we no longer need to think about doing them. And although they may seem small, they are an incredible feat of the brain, because by making these routine behaviors automatic, it frees up so many cognitive resources that can be used for other tasks. Because, I mean, could you imagine having to put the same amount of thought and effort into driving as you did when you were first learning how to drive? It'd be a lot. I mean, it wouldn't be the worst thing. I'm from New York, so I know some of those drivers could use that. But yeah, so we have this really powerful system in our brain that is guiding our behavior throughout our day-to-day lives. However, this system can be hijacked by drugs of abuse, and is one of the reasons why addiction is so difficult to treat. Because once someone becomes addicted to a drug, he will seek and take it as automatically as you and I turn on that light switch. And now, the automation of drug seeking and taking behaviors is one widely accepted model of how addiction develops. And it states that when someone is first abusing a drug, they take it because it's rewarding. They take it in order to get this reinforcing outcome. And in this sense, the taking behavior can be considered to be under goal-directed control. Again, the behavior is performed in order to obtain that outcome, to obtain that goal of getting the reinforcing feeling. And as the individual continues to take the drug over time, the brain makes the same shortcut as it does when we're practicing how to drive or turning on those light switches. It makes these drug taking and seeking behaviors automatic. And at this point, the individual is considered to be addicted, because here the behavior is no longer directed in order to get the outcome, it's done automatically. And so this is why people suffering from addiction will continue to take a drug even if they no longer want to take the drug, even if it's harmful to their health or their family. Again, the element of choice is removed and this behavior has become automatic. And so the ability to mediate behavior, the ability to automate behavior is mediated by a part of the brain called the dorsolateral striatum, which is a part of subcortical nuclei that integrate cognitive, sensory, and emotional information in order to guide our behavior. And in particular, within the dorsolateral striatum, the formation of automatic behavior is associated with strengthened signaling of the neurotransmitter glutamate. And so this has been studied in automatic behaviors involving food rewards and learning tasks, but it hasn't yet been looked at in the context of addiction. And so to answer this question, we need to take a closer look at how glutamate signaling works. And so here's a quick overview. In our brain, we have billions of brain cells or neurons that communicate through releasing neurotransmitters like dopamine or serotonin. And so if we zoom in to this point of communication, we'll see, hopefully not cartoon version in our brains, but we'll see this first signaling neuron, a second receiving neuron, and the space between the two is called the synapse. And we also see another kind of cell that's enveloping the synapse. It's called an astrocyte. And these cells perform a lot of supportive functions for neurons and participate in neurotransmitter signaling. Okay, so we have our signaling neuron and it's releasing glutamate into the synapse, where it then binds onto receptors on the receiving neuron. And this generates a signal in that neuron, which is then passed on to the next neuron and the next and the next. But this signaling can't go on forever. So glutamate is gradually removed from the synapse by a class of proteins known as glutamate transporters. And these glutamate transporters are critical for proper glutamate signaling because by removing glutamate from the synapse, they control the concentration of synaptic glutamate as well as the time course of glutamate in the synapse, which influences the strength of the overall glutamate signal. And these transporters come in several different subtypes and are expressed on neurons and astrocytes, but I'm particularly interested in the glutamate transporter 1, or GLT1, because it's the most predominant glutamate transporter. It's mostly expressed on astrocytes, and it accounts for approximately 90% of glutamate clearance in the brain. So you can imagine if something were to happen to this glutamate transporter 1 system, it would have large ramifications on glutamate signaling. And we've observed that this is the case in the context of addiction. And so several studies have shown that rats with a history of cocaine self-administration display decreased GLT1 expression and glutamate uptake capacity in the ventral striatum. And this is an area of the brain that's involved in encoding the rewarding aspects of drugs. In addition, these decreases to the GLT1 system are associated with strengthened glutamate signaling within this region in addiction. Again, because if you're having decreased glutamate transport, glutamate is going to be in the synapse for a longer period of time. So it can continue to bind onto receptors on other neurons as well as presynaptic neurons and affect the strength of the glutamate signal. And so everyone's been focused on the ventral striatum for a very long time because it's a very hot topic in the rewarding aspects of drug abuse. But finally, in 2016, Ducre's group looked at the dorsolateral striatum, and they observed that GLT1 expression is decreased in the dorsolateral striatum. And this finding opens up a lot of different doors. First, it raises the question, is the function of GLT1 altered? In addition, are differences in GLT1 expression and function related to the different classifications of drug-taking behaviors, such as goal-directed or automatic behaviors? Is this disruption to the GLT1 system, could it be the link between the strengthening of glutamate signaling in the dorsolateral striatum that's needed to automate these drug-taking behaviors? And we think so. So we hypothesize that rats with automatic cocaine-seeking behavior will have reduced glutamate, GLT1-mediated glutamate clearance and expression in the dorsolateral striatum as compared to goal-directed cocaine-seeking rats and saline controls. And so the first step in addressing this hypothesis is to generate rats that either seek cocaine in a goal-directed or automatic manner. And to do this, we train rats on a chained cocaine self-administration paradigm. And what's really great about this paradigm is that it incorporates both drug-seeking and taking behaviors, which provides a more comprehensive assessment of addiction. And so here's how the paradigm works. First, we train rats to self-administer cocaine. So we place the rats in a training chamber where they learn that when they press the taking lever, they are given an infusion of cocaine. And once the rats have learned this, we then make it a little more complex. We introduce a second lever, the drug-seeking lever. And so here the rat needs to learn that when they press the drug-seeking lever, it gives them access to the drug-taking lever. And when they press the drug-taking lever, it gives them an infusion of cocaine. And so here's where the idea of the chained behavior comes into place. It's kind of two links in the chain. The first link is between the seeking lever and gaining access to the taking lever. And the second link is pressing the taking lever and getting cocaine. And this way, we're able to look at both seeking and taking behaviors separately. And so the rats train on this chain schedule for several weeks till they've really learned to seek and take cocaine. Then we can assess whether this cocaine-seeking behavior is automatic or goal-directed. And the first step in doing this is to take away the value of the taking lever. And we call this devaluation. So here the rat is put in the training chamber, he sees the taking lever. However, no matter how many times he presses it, he no longer gets an infusion of cocaine. And so the rat will do this for 2 weeks until he's reached the point that this taking lever no longer has its value. It no longer gives him the cocaine that he wants. We then ask, does devaluation of the taking lever affect cocaine-seeking behavior? And this can give us insight into whether or not the rat is abusing or addicting cocaine. And so we do this through a series of 2 tests. In the first test, we measure the seeking lever presses the rat makes, and this is considered to be under devalued conditions because the rat has just come out of devalued training. In this test, the seeking lever presses have no responses. We're just interested in how much they are going to seek out that behavior. And so after that, we then revalue the taking lever for 2 days, where the rat goes in, but this time when he presses the taking lever, it gives him cocaine, it gives him that response. And then we repeat the same seeking test, but now this is considered to be under valued conditions, again, because the taking lever has its value again. And so by comparing the number of these seeking lever presses, we can compute what is called the cocaine seeking score, which is the number of seeking lever presses made under devalued conditions expressed as a percentage of those made under valued conditions. And these cocaine seeking scores can tell us whether or not the rat is abusing or addicted to cocaine. And so in the case of abuse, again, the cocaine seeking behavior is goal directed. The rat is pressing that seeking lever in order to gain access to the taking lever. But if we take away the value of the taking lever, then the rat is going to decrease the number, it's going to decrease his seeking for it, because again, this whole behavior was geared towards getting that outcome. And so this will be reflected as a low cocaine seeking score. In comparison, in the case of addiction, this behavior is considered to be automatic. So again, the rat will press the seeking lever regardless of the outcome. So it really doesn't matter whether or not the taking lever has its value, the rat will continue to press the seeking lever. And this is represented as a high cocaine seeking score. And so when we look at the distribution of cocaine seeking scores with all of our rats, we see this really great spread. The mean cocaine seeking score of the group is around 100. And so we've defined cocaine seeking scores above 100 as automatic cocaine seeking. And we see about 47% of those rats are displaying that behavior. And below 100 is goal-directed cocaine seeking or abusive behavior. And about 53% of our rats are displaying that behaviors. So this suggests that this chained cocaine self-administration paradigm is a really great way to assess these 2 different modes of behavioral control. And so now that we have those rats, the next step is to look and see what's changing in the brain and what's different between these 2 groups of rats. And in particular, I'm interested in visualizing glutamate clearance within the dorsolateral striatum of goal-directed and automatic cocaine-seeking rats, as well as yolked saline controls with a technique known as igloo sniffer. And so glue sniffer stands for the glutamate sensing fluorescent reporter, which essentially consists of a modified green fluorescent protein that when glutamate binds to it, it generates a fluorescent signal. And what's really great about this reporter is that it can be expressed on either astrocytes or neurons and is tethered to the extracellular membrane. And again, since we're interested in the role of astrocytes in addiction, we expressed glue sniffer on astrocytes. And so first we inject glue sniffer into the dorsolateral striatum of rats and express it on astrocytes. Then the rats undergo training on the chained cocaine self-administration paradigm. And after that, I collected slices of their brains that contained the dorsolateral striatum and conducted glue sniffer imaging. And so here is a close-up picture from my glue sniffer imaging. This is in the dorsolateral striatum. And so this is really zoomed in for reference. This is my stimulating electrode, which is the size of about a couple of hairs. And so you can see that here we have our astrocytes that are expressing glue sniffer in green and yellow. And so what I'll do is that I'll take the stimulating electrode and I'll use it to stimulate neurons in the striatum with a 1 milliamp pulse. This causes the neurons to release glutamate, which then binds onto our astrocytic glue sniffer to generate an increase in fluorescence. And I call these signals evoked glutamate glue sniffer signals. And so I can then measure this evoked glue sniffer signal within this region of interest here, and we get this great representation of that trace. So here we're looking at how the delta F over F changes over time. And what delta F over F is essentially the change in fluorescence that's corrected to the background fluorescence. And so here again, you can see we have our baseline glutamate, our stimulation induces a robust increase in fluorescence, which decays over time as glutamate is being cleared out from the synapse. So we were really excited to get these great glue sniffer signals, but we first needed to make sure that these signals were in fact coming from the stimulation of neuronal glutamate release. And so to test this, we bath applied tetrodotoxin, or TTX, which is a toxin from pufferfish that inhibits the release of neurotransmitters. We then delivered the same 1 milliamp stimulation and recorded the trace. And so in comparison to the trace I just showed you before, where we get this great increase in fluorescence after stimulation in the absence of TTX, when we bath apply TTX for 15 minutes and we stimulate, we don't see an increase in glue sniffer fluorescence. So all in all, this suggests that we are in fact measuring glutamate that is being released from neurons following stimulation. Okay, so coming back to our main question, is there a difference in glutamate clearance between rats that are abusing, addicted, or naive to cocaine? And to answer this question, I'm interested in this portion of the traces where the fluorescence signal is decaying. Again, because this represents the fluorescence signal as it decays, it represents glutamate being cleared away from the synapse. And in particular, when we fit a nonlinear regression to this part of the trace, we're able to compute the decay tau, which is a rate constant of glutamate clearance. And so if something has happened to our glutamate transporter system, it will be reflected in the decay tau as a longer time for glutamate to be cleared away from the synapse. And so we're about halfway through this study, and here are our preliminary results. When we compared the decay taus of evoked glutamate signals in the dorsolateral striatum between saline rats and cocaine and rats that self-administered cocaine, we didn't find a significant difference between them. However, when we separated the cocaine rats out into their behavioral classification, either goal-directed or automatic cocaine seeking, we did find the difference. First, in comparison to the decay taus of signals in saline rats, we found that the decay taus of goal-directed rats didn't significantly differ, which is what we hypothesized, again, because those behaviors haven't become automatic yet. However, in rats that automatically seek cocaine, we found that their decay taus were significantly longer than those of saline rats, which suggests that glutamate is taking a longer time to be cleared away from the synapse and that there could be an impairment within our glutamate clearance system. We did not, however, find a significant difference between clearance in the goal-directed and automatic rats, although there was a slight trend. And so it'll be interesting to see if this effect becomes significant when we analyze the rest of the rats. So taking all this together, what does this mean? So first, we can conclude that the chained cocaine self-administration paradigm generates both goal-directed and automatic cocaine-seeking rats. So it's a really great and reliable platform for investigating the different modes of behavioral control and addiction and their neural correlates. And second, we also found that rats with automatic cocaine-seeking behavior have impaired glutamate clearance in the dorsolateral striatum, which is exciting because this is what we hypothesized. And so this raises several other questions. First, is there decreased function of the glutamate transporter 1 in particular? And future experiments in the lab will address this through using glutamate transporter 1 antagonists. And the next question is, is the expression of GLT1 within the dorsolateral striatum decreased, as we've seen in similar studies. And current experiments in the lab are underway to address this with Western blot analysis. And so these results are really exciting because they are the first step in developing a more comprehensive approach towards treating addiction. Because if we can understand the specific changes in the brain that are happening, we can develop and identify new biological targets for treatment. We can then use this knowledge to develop more effective medications and combine them with more refined behavioral treatment strategies, bringing us one step closer towards helping individuals overcome this disease. And so with that, I'd like to thank my lab and our collaborators within the Wilcox lab as well as my committee within the neuroscience program and the directors and organizers. And last but definitely not least, our funding, in particular, the Skaggs Graduate Research Fellowship. I am really grateful to have been recognized through this award, and it has supported me this past year, and I've been able to do a lot of great things, go to several international conferences to complete that Science Communication Fellowship. So I am just very, very grateful for this opportunity and all that it's provided. And so with that, I will take any questions.
Maybe I can ask a question, Daniel. This
Sure.
is Gandhari. Just a couple of questions, and this is outside of my field, but how do you correlate the molar concentration of this clearance of the compound that you were talking about, the molar concentration with the intensity of imaging? That's one question. The second question is, what is the correlation, or what is the known correlation between rats and humans in this behavior?
Those are great questions. And there's a lot that goes into the kinetics of glue sniffer imaging. And so the work that's been done with this specific reporter in terms of its clearance abilities, it's been shown to be very similar to clearance abilities of the natural GLT1 transporter. And I guess most of that work, to answer your question, is done, it compares glutamate clearance to other assays like tritiated glutamate uptake or slow transporter currents. And so the glue sniffer is very sensitive to glutamate release. So I'm guessing your question is on the molar concentration of iGlue sniffer or glutamate?
Yeah, sometimes images just don't give you the quantification that you want. I was just curious, how is the assay in terms of quantification?
Quantification, yeah, that's a great question. And so, when they were developing this glue sniffer tool, they did side-by-side electrophysiological comparisons, and they found that those kinetics were the same. But again, it can't be a perfect system. Something that we are working on in the lab is to look at synaptic slow transporter currents within astrocytes. So that's going to provide us a way to look at how glutamate is going to be cleared within the astrocytes without the presence of glue sniffer as well. But yeah, there are a lot of factors. And so your next question was in terms of how the behaviors in rats correlate to humans in terms of the addictive and cocaine seeking behaviors? Just like if we observe that, or?
There's always this question of animal to human correlation, right? In type of drug studies. I was just curious, how is it with your types of studies?
Yeah, and that's a really great question. The emergence of adding seeking behaviors into the animal models actually has been a recent one. And it's you know, when we think about human seeking behavior, it's not going to be as simple as pressing a lever. It is going to be more complex. And so that's why by having this kind of sequenced behavior, we're able to look at seeking versus taking behavior. Studies within humans suggest that there are different neurocorrelates between kind of seeking and taking behaviors, and that on the seeking behavior side is more of a chain of behaviors. And so if we can understand that chain, then it'll help us kind of understand that stage of behavior as well.
Great, thank you. Great presentation.
Thanks.
Any other questions for Danielle? Okay.
I have one, but I'll put it in the chat.
Okay.
I think our next introduction will be by Dr. Lim. Do we have Carol on?
You know, I don't think she's on.
Okay.
Yeah.
Why don't we skip to Eric introducing Snigdha?
Okay, no problem. I'm just waiting for my video to come on here. All right. Okay, thanks, Randy. And so next up is Snigdha Sarkar. Snigdha joined us from the Indian Institute of Technology in Delhi, which is one of the top universities in India and one of the great universities of the world, where she performed very well and ranked nationally in chemistry. And we were lucky enough to bring her here to the biological chemistry program at the university and then to my laboratory in the Department of Medicinal Chemistry. There she's been involved in a variety of projects focused on understanding how peptides can be decorated en route to our goal of gaining control of peptide synthetic biology. In which we're using in a variety of ways, looking at different drug targets. Snigdha has so far published or submitted 3 papers in this area, which are very strong. And in addition to her strong research work, one of the things that I most like about her is that she's been a great mentor and role model within my lab. And so she's helped many, many people with their problems. She's soft-spoken, and really keeps things calm. She's been in charge of our mass spectrometry instruments, which is a huge workload and involves interfacing with many students in the department and the college. And so I think her mentorship has also been very strong. In addition, she's been a student leader in a variety of committees, important committees around campus, including participating in very important task force in our program to revise curriculum. And so with that, I will leave it to her.
Okay, so thank you for the kind introduction, Eric. So today I'm going to talk to you about a macrocyclase that we characterized in the past year with support from the Skaggs Fellowship. Cyclic peptides have emerged as a prominent drug class for several years, as they can combine several advantages of small molecule drugs with those of biologics. Shown here are some examples of widely used drugs that have a peptide backbones. You can see that they have varied biological and pharmacological properties, ranging from anti-cancer to antibiotic activity. Due to their benign framework, cyclic peptides have very low toxicity. In addition to this, their rigid conformation make them a stronger binder to their target, make them more permeable, and also highly bioavailable, as they can resist proteolytic degradation. There are several ways in which you can make cyclic peptide and cyclic peptide libraries, both chemical and enzymatic. But all methods suffer from some inherent disadvantage, like the use of expensive rare earth metals, or the problem in carrying the reactions in vitro, or just the sheer entropic loss associated with cyclization reactions that make scaling up quite difficult. These factors together leave room for catalysts that can catalyze the cyclization of a wide variety of substrates efficiently. So we wanted to use the resources in our hand involving cyanobactin biosynthetic machinery to tackle these problems. Our lab has worked on cyanobactin biosynthesis for over 10 years. Cyanobactins are peptide-based natural products isolated from cyanobacteria, which are synthesized at the ribosome during translation and then heavily modified by several biosynthetic enzymes. So the peptide backbone is genetically synthesized by the gene E, and then the backbone can be modified either by the addition of alkyl chains to selective amino acid residues or by the incorporation of azolene rings into the backbone. However, the common feature amongst most cyanobactins is that they are cyclic in nature. Just like the other modifications, the cyclization reaction is also carried out by one of the biosynthetic enzyme, which is encoded by the gene G. The Cyanobactin macrocyclases are dual function enzymes. They act as both a serine protease as well as a macrocyclase. So these enzymes have a unique helix-turn-helix motif at the active site that bind to a recognition element in the substrate called RS3 and place them in the vicinity of the active site serine. Thereafter, the serine attacks the substrate to release the recognition sequence 3 and leaving the substrate covalently captured at the active site. After the proteolysis part is complete, the free N-terminus of the substrate attacks the acyl enzyme intermediate to form the cyclic peptide. It's important to note here that the recognition element, which is recognition sequence 3, leaves the substrate in the course of the reaction and is not part of the final product. This quality makes Cyanobactin macrocyclase extremely tolerant of wide substrate changes. In other words, as long as the recognition sequence 3 is present, any natural amino acid containing peptide would be cyclized by the enzyme. Other than the presence of recognition sequence 3, the other factor that is required for successful cyclization for all previously characterized macrocyclase is the presence of a thiazolene ring at the ligation juncture. This thiazolene ring provides a conformational rigidity to the peptide backbone and helps the N-terminals attach to the enzyme. However, you need another biosynthetic enzyme from the biosynthetic pathway to install the thiazolene ring. As a result, it complicates the downstream translation of the macrocyclase to practical applications like generating a library of peptides. We wanted to find an enzyme from cyanobactin pathways that has all the advantages of these macrocyclase, like very flexible substrate tolerance, but is not dependent on the presence of a thiazolene ring. We collected several Cyanobactin macrocyclase enzyme sequences and analyzed them through this phylogenetic tree. At this point in the study, only 3 enzymes from this family had been biochemically characterized, but we were able to assign the function of each of the enzyme by matching them with the final cyanobactin isolated from their pathway or by looking at their precursor peptide sequence. So in this representation, each gray circle represents the amino acids in the native cyanobactins that are present at the different positions, and the red circle represents the site of macrocyclization. This analysis provided us with a cheat sheet or a map where we could choose the enzymes according to the products we wanted to make. You might notice that most of the red circles are occupied by cysteine. These cysteines are converted to thiazole or thiazolene before the macrocyclization could actually take place. However, at the places where the cysteine ring is not present at the ligation juncture, it is replaced by a highly conserved proline. Therefore, we focused our efforts on finding a Cyanobactin macrocyclase that is proline specific. Particularly, we focused on one belonging to Prenylagaramide biosynthesis, which also has a conserved proline ring at the macrocyclization site. Prenylagaramides are cyanobactins isolated from Planktothrix agarthi in 1999, and the biosynthetic pathway involved in its production was sequenced in our lab in 2011. The first step in the biosynthesis is the generation of the precursor peptide at the ribosome. Each precursor peptide contains a core, shown in blue here. As the name suggests, the core peptide is what makes the core or the backbone of the natural product. So after the ribosomal formation of the peptide backbone, an enzyme cleaves the N-terminus of it and frees up the amine group at the N-terminus. This leaves us with the core peptide appended to a conserved recognition sequence at the end. So we can see that there are three different precursors here, although only precursors 1 and 2 form natural products. Any peptide similar to the sequence in peptide 3 was not isolated from from culturing the cyanobacteria. We expect that the next step in the biosynthesis is the macrocyclization reaction, where the enzyme PagG would cleave off the conserved recognition sequence and cyclize the rest of the core peptide to form the cyclic peptides. If PagG behaves like the previous members of this family, we can expect that as long as the conserved recognition sequence is present, any other core peptide sequence can be appended before it for it to undergo successful cyclization. As long as the recognition sequence is conserved and the C-terminus proline is present in the peptide. The last and final step in biosynthesis is the addition of isoprene groups to selective tyrosine residues in the cyclic peptide to form the final natural products Prenylagaramides C and B. So we started with biochemical characterization of the enzyme PagG. For that, we encoded the gene for the enzyme in a PET28B vector and expressed it as a histagged construct. We purified the expressed protein using nickel affinity chromatography and size exclusion chromatography. Shown here is the SDS page analysis of the purified protein, which was used in all further in vitro reactions. To test the macrocyclization activity in vitro, short substrates that mimic the native substrate of the enzyme were incubated with PagG for overnight at room temperature. So the substrate contained the conserved recognition sequence and the different core peptides. We monitored the progress of the reaction by LCMS. Both substrates 1 and 2 underwent successful cyclization. As we can see here, at 0 time point or at the beginning of the reaction, the product peak signified by compound 4 here is not present. However, after overnight, with time, we can see that the product starts accumulating in presence of PagG. As in the native cyanobactin pathway, substrate 3 does not undergo cyclization in presence of PagG. Therefore, we could conclude that our enzyme was following the natural behavior in cyanobacteria. Taking together, these data represent the first evidence of a proline-specific macrocyclase from a cyanobactin pathway. Next, we wanted to recapitulate the pathway in vitro, and in doing so, we treated the cyclic peptides obtained in step 1 with the prenyltransferase and putative isoprene donor dimethyl allyl pyrophosphate. Both substrates 1 and 2 went to the final natural product formation of Prenylagaramides C and B. We can see it again through LCMS with the appearance of the peak for Prenylagaramides C after overnight incubation with the prenyltransferase. Also, any linear peptide that remained unreacted after step 1 also underwent prenylation in presence of the prenyltransferase to form product 8. With the help of the enzymes PagG and PagF, we were able to present the first total synthesis of Prenylagaramides C and also were able to synthesize Prenylagaramides B. Next, we wanted to investigate the substrate tolerance of this enzyme using a saturated mutagenesis library based on the wild-type precursor as shown here. In this peptide library, each position of the core peptide was mutated with 20 proteinogenic amino acid. But in each of the peptides, the macrocyclization site, proline, and the recognition sequence remained the same. We needed the proline and the recognition sequence constant so that they could dock into the capping helix and cyclize the core peptide. This peptide library comprised of a total of 120 peptides, where 114 were unique peptide sequences, and there were 6 repeats of the wild-type peptide. The different members of the library were arrayed into multiple different wells and treated stepwise with the macrocyclase PagG and the prenyltransferase PagF. The reactions were monitored with LCMS, and the yield for each peptide was calculated with calculated as the area under the curve of the product peak, which was normalized with respect to an internal standard. Here we present our findings in the form of a heat map. So in this heat map, each box represents a different peptide. The columns represent the different proteinogenic amino acid, and the rows represent the core peptide positions. So for example, the first box in row 1 is ANPYLYP FHEDDAE. The second box is DNPYLYP and so on. We decided to take out cysteine from our studies because they formed non-specific polymeric products due to disulfide bridges. So in this heat map, all the yellow and greenish boxes are the peptides that were accepted by the macrocyclase, and the blue boxes are the ones that were not cyclized by our enzyme. For example, any peptides containing aspartate or glutamate residues were not favorable substrate for the enzyme. Also, positions P7, P2, and P3 had much stricter substrate preference than the middle positions. The peptides the cyclic peptides that we obtained after reaction with PagG served as the substrate for the prenylation reaction. So these peptides were treated with PagF and the isoprene donor, and the results are again presented in the form of a similar heat map. All the gray boxes in these heat maps are the peptides that were not formed in the first reaction and hence are nullified for this study. And then the blue boxes and yellow boxes signify peptides that were not formed and the peptides that gave prenylated products. We noticed for PagG reactions that they had much relaxed substrate tolerance, and as long as the native tyrosine was present at the P4 position, most of the peptides were accepted by this enzyme. Here we represent our findings in a pictorial fashion where each of the circles are denoted with the amino acids that are accepted at that particular position. With these results, we were able to discern some substrate rules for our enzyme. For example, for PagG, any peptides containing the acidic residues glutamate and aspartate were not accepted, and this is likely because of incompatibility with the highly acidic recognition sequence. Moreover, the internal positions were much more forgiving than the terminal positions. We can draw on the knowledge obtained from the crystal structure of a thiazoline-specific enzyme PagG with its substrate, which was published in 2012. According to that study, the middle residues had much lesser van der Waals interaction with the protein surface than the terminal residues, which is likely the reason why the terminal ones have much stricter substrate tolerance. For PagF, we noticed that the substrate rules were much more relaxed, and as long as tyrosine is present in its native position, the other positions can accept almost any other amino acids. Both the substrate rules for PagG and PagF would help us or any other user in deciding whether these enzymes would work for synthesizing the products they want to make. So when we looked at the different enzyme sequences, we noticed that the capping helix sequence is followed by an internal recognition sequence embedded within the enzyme. This embedded recognition sequence were very similar to the RS3 sequences in the core peptides. These observations prompted us to propose that the enzyme remains in an internally deactivated state in the absence of the substrate, where the capping helix binds the internal recognition sequence. However, when the substrate is added to the enzyme in excess, it replaces the internal recognition sequence with its own recognition sequence, which then undergoes transamidation reaction to form the cyclic peptide. If this hypothesis is true, if we replace the internal recognition sequence, then the recognition sequence in the substrate would also change consequently. To test our hypothesis, we took the sequences of our proline-specific enzyme PagG and the thiazoline-specific enzyme PagG and formed a series of chimeric enzyme constructs where the capping helix and the embedded recognition sequence were either swapped or added onto the enzyme. We tested these constructs with the wild-type substrate as well as hybrid substrate, but none of our constructs were active with the substrates we used. So this is still an area that we are actively working on, and we will try to figure out if we can construct better chimeric enzymes that would be active and help us test the hypothesis. So to conclude, we presented the first example of a proline-specific macrocyclase and used a macrocyclase as well as the prenyltransferase in conjugation to synthesize the natural products. We also used the same strategy to synthesize and analyze more than 300 peptides within 2 days from a starting point of 120 peptides. In the future, we would like to engineer better chimeric constructs, which would help us test this hypothesis. And a long-term goal for this project is to use our strategy for in vitro drug testing. With that, I would like to thank a lot of people, most importantly, my PI, Dr. Eric Schmidt, who is always available with a lot of helpful suggestions, and all members of my lab who kept the lab running smoothly even in these trying times, and most importantly, Venja, who worked on this project before me and taught me most of the techniques used in this study. I am very grateful to the ALSAM Foundation and Skaggs Graduate Research Fellowship, who played a crucial role in accomplishing this project. With that, I can take any questions.
Okay, thank you, Snigdha. Any questions?
I have one.
Go ahead.
Oh,
Go ahead, Randy.
Oh, I was just, I thought that the saturation mutagenesis study was really interesting. Many of the tolerances were predictable. So for example, if there was a hydrophobic residue, the ones that were hydrophobic substitutions were acceptable. But there were some exceptions, such as glutamine being acceptable in the P7 position. And have you been able to learn anything from the exceptions to what would be expected?
So like the study was carried out for hexapeptides. So if, for example, someone had to use this enzyme for their substrate, they might want to avoid glutamine or proline at, say, P7 position. So I'm not completely sure why similar amino acids would be, one would be acceptable and the other would be not. Probably if we can find the crystal structure of the enzyme, then we would be able to say it for sure why certain amino acids are acceptable.
Great, thank you.
So Snigdha, just to kind of follow up on that, and it kind of crystallizes my question. Let's say we had let's say for some reason that glutamine at P7 is not acceptable. Well, is it possible that a glutamine at P7 could be compensated by a mutation somewhere else? And using this strategy, you can't really find out if there are pairwise or three-way kind of compensation. You can't really look at all the diversity of the sequences. And so is there a way that you're thinking about, or is it technically possible to come up with an incredibly diverse library where you can look at the full sequence diversity of these systems, where you look at sort of, all combinations and maybe take a genetic approach or something like that to try to expand this diversity? Do you understand my question?
Yeah, that's a really good point. So yeah, we took a really small library for our first in vitro testing. However, if we were to expand on this, we could use the enzyme and embed it in a vector and use NNK library in the precursor that would perhaps be able to help us test even larger variations in the precursor.
Yeah, I'm thinking in the drug screening context, that might be really important, because you might find something where a glutamine at P7 is really important, and if you compensate with an aspartate at some other position, then that would give you an active compound, whereas this sort of kind of natural sequence driven approach doesn't allow you to do that.
Yeah, got it.
Okay, thank you. Beautifully done, Snigdha. We'll now ask Dr. Lim to introduce Erica.
Hi, it's my pleasure to introduce my graduate student, Erica Vandermouse. She received her BS in chemistry from University of Wisconsin, Oshkosh in 2015 and joined our department in my lab in 2016 through the BC program. She came up with a new project and started a collaboration with the Atanakovic lab centering on cancer immunotherapy, and has worked in a large fraction of her time with Tim Lutkins. She's currently a co-author on 5 publications, including a Nature Communications paper, and has several first author papers forthcoming. In addition to the Skaggs Fellowship, she's also had an AFPE pre-doctoral fellowship and a Ralph and Clara Rutledge Memorial Scholarship, and has served as SAC on our department and heavily involved in the BC program through curriculum advising and admissions committee. And so I'm happy to have her give her talk that she will present today. Thank you.
Okay, so before I begin, I just wanted to thank the LSAM Foundation for this Skaggs Fellowship that made all this research that I'm about to present here today possible. I'm going to be looking at improving CAR T-cell efficacy and safety, and looking at that, I'm going to be using ovarian cancer and multiple myeloma. So first, just a little bit about the CAR T-cell therapy. We can see here a CAR, or a chimeric antigen receptor, the structure shown over here. Importantly, it's a modular protein that combines antigen recognition and also T-cell signaling into one single protein. And we can see here on the extracellular portion of the CAR, we have an scFv followed by several other domains that confer cytotoxicity and also signaling into the T-cell to promote proliferation and also survival. So after the CAR is constructed in the patient, we have the T-cell isolation from the patient, where we isolate the normal T-cells, and then we use the lentivirus to transduce our CARs into the T-cells and expand them ex vivo or in the lab until we get enough of the T-cells that we have a therapeutic level that we can transfuse back into the patient. CAR T-cell therapy recently has been very successful for B-cell malignancies. In 2017, there was 2 approvals of Kymriah and Yescarta that target CD19 and B-cell malignancies. Kymera and Yescarta both improved the survival for patients who were previously didn't have good survival and were relapsed and refractory to other forms of treatment. And so this really gave hope that we could maybe use this in a lot of other types of cancers. But so far there hasn't been other approvals. And part of that is because of lack of ideal CAR targets available for other cancers. And 2 of the problems that we're going to look at today are heterogeneous expression of the targets on tumors, and also tumor-associated antigens that are targeted that are expressed very highly on malignant cells, but also at lower levels on healthy tissues. And so first we'll look at the efficacy and tumor heterogeneity in ovarian cancer and how we might be able to overcome that. Ovarian cancer has a very poor prognosis as the most lethal gynecological malignancy with less than 50% 5-year survival rate. There are many CARs currently being developed and in clinical trials for ovarian cancer, and one of the top candidates is folate receptor alpha, and so we're going to be looking mostly at that one today. And 70% of patients test positive for folate receptor alpha. And so that sounds like really good, and like most of the patients would be able to benefit from this therapy. However, when you look at the staining for folate receptor alpha on individual tumors, you see the heterogeneity come into play. Of those 70% of patients who test positive, they fall either into this low, medium, or high category. And looking at these here, the brown cells are positive for folate receptor alpha, and the blue are negative. And clinical trials typically call for patients that are either medium or high for folate receptor alpha. And so that means that up to 50% of the cells in the tumor aren't going to be actively targeted by the CAR T-cell therapy. And so I made a cartoon to show this a little bit more clearly, where we have our heterogeneous tumor again, and then the CAR T cells are only going to actively target the cells that express the receptor, which will leave behind a lot of other malignant cells that don't express the receptor, and those will be prime candidates for recurrent disease and won't lead to very durable responses potentially. And so looking at this, it'd be great if there was a way to target the cells that are in close proximity to our antigen presenting cells. even if they don't express the target, because it's a solid tumor, they're all going to be very close. And T cells already have a way to do this through the CD95 pathway. So CAR T cells, after they bind their target, kill the target cell through this granzyme perforin B pathway. But in addition to that, they also upregulate the CD95 ligand on their cell surface. And then they can bind CD95 on cells that are nearby and confer cytotoxicity to those cells as well. However, in cancer cells, this pathway is almost always inactivated. And so right now, the CAR T cells can't take advantage of this mechanism of cytotoxicity. So again, if we're looking at the cartoon, we can see here that this time, if we have the heterogeneous solid tumor, that any cells in proximity to the cells that the CAR T cells bind could also potentially be killed through this other pathway and then result in more durable responses for patients. So we're going to look at then the CD95 pathway in ovarian cancer cells. We see here that we have the receptor present on our ovarian cancer cell lines, SKOV3, OVCAR3, and Kuramochi. And this Jurkat cell line is a T cell line that we're using as a control for cells that have an active CD95 pathway. So even though we have this very positive staining of the CD95 receptor on the surface, when we use an agonist antibody to confer cytotoxicity through this pathway, we see that even though in the same conditions that we see this big increase for the Jurkat cell apoptosis through the CD95 pathway, we don't see any significant difference in our SKOV3 or our Kuramochi apoptosis, and we see only a small increase in OVCAR3, showing that the CD95 pathway is indeed not active in these cell lines. And so in order to restore the CD95 pathway, we have to look further than the receptor, because there must be something other than the receptor that's not functioning in these cells. And so just very quickly, the CD95 pathway, after the ligand binds the receptor on the surface, there's a conformational change that allows FADD to be recruited to the intracellular portion of CD95, which then recruits caspase-8, forming this DIS complex. The DIS complex is very important as it brings caspase-8 into close proximity to another caspase-8 and allows it to autocatalyze into the activated form of caspase-8. Prior to its activation, it cannot carry forward with apoptosis, but once it's activated, depending on the level present in the cell, it can either go through this extrinsic pathway of apoptosis, which is more direct, or if there's less caspase-8, it can still confer apoptosis through the mitochondrial pathway or the intrinsic pathway. Our goal is to try to restore the pathway through this intrinsic pathway, as there's a lot of known mechanisms of resistance in ovarian cancer to this intrinsic pathway of apoptosis. So our hypothesis is then that caspase-8 gene therapy will restore the CD95 pathway in ovarian cancer cells. And we were able to restore the pathway in one of our cell lines. And we see here the SKOV3 cell line, that when we transduce caspase-8 into the cell and then either treat with the isotype control antibody or this agonist APO1 antibody, that we significantly increase the apoptosis that we see for the APO1 and the caspase-8 above our control. And importantly, the caspase-8 on its own doesn't kill the cells, because we really don't want the caspase-8 to kill the cells. We want the combination of the CAR T cells being in close proximity to kill the cells. And we also see significantly more cell death compared to our control, which is an EGFP transfection. And EGFP shouldn't affect the CD95 pathway. And so this is also significantly more. So now that we have restored the pathway, at least sensitized the pathway somewhat, we looked at the folate receptor alpha expression on the SKOV3 cell line so that we can use the CAR T cells against them. Unlike a patient tumor, cell lines are very homogeneous as they're from a single cell. And so we see here, as we'd expect, very high staining on all of our SKOV3 cells. But this doesn't accurately represent a tumor like that we saw in those histograms earlier. And so we are introducing the heterogeneity of the folate receptor alpha staining using CRISPR. And so on day 0, we did an RNP liposuction to introduce the entire CRISPR-Cas9 complex into the cells. And then we wait several days for the Cas9 to knock out folate receptor alpha at the genomic level, and then also for that to be reflected at the expression, and so that we can just use cell sorting to stain for folate receptor alpha and then select this very negative population of cells into a single well and allow those to grow up, and then stain for folate receptor alpha and also confirm the knockout with sequencing. And so if we see here, we have the folate receptor alpha staining. Here's our negative control isotype. And so we got some of the cells that had the double knockout in both alleles for folate receptor alpha and don't express anything on the surface. We also have this median expression that we confirmed through sequencing that one of the alleles was knocked out while the other one was unaffected. And then we have our original or our unaffected cells that have this high expression for folate receptor alpha. And so now that we have these cells, now we can go into looking at the CAR T cell efficacy against the folate receptor positive, medium, and negative cells, and also create the co-cultures of the folate receptor alpha positive cells and negative cells and transduce with caspase-8 followed by CAR T cells to see if our hypothesis is correct that we can increase the killing of the folate receptor negative cells that are in close proximity to the folate receptor positive cells. If that's possible, we would like to go back and try to restore the CD95 pathway in the other cell lines we have, and ideally find a single agent that would restore the pathway in all of the ovarian cancer cell lines, and then move forward with in vivo modeling of this. So this is really looking at a tumor heterogeneity, but another problem that we see with CAR T cells is the safety due to low expression of the target antigens on other tissues, other healthy tissues. And so for this, we're going to look at multiple myeloma. Multiple myeloma is a hematological malignancy, and CAR T cells have been tried in clinical trials before, but the results aren't good enough, and they're not durable, and they don't last long enough to warrant an FDA approval of these targets. And so the Lutkins and Adanakovic lab identified a target called CD229 that's highly expressed on multiple myeloma cells, both newly diagnosed multiple myeloma and relapsed refractory. And so that's important so that more patients will be eligible for this type of therapy. So now after they identified this target, they also developed a fully human scFv against CD229 and used that to make a CAR T cell. And so here we're showing the scFv is called 2D3, and we're showing the efficacy of these CAR T cells against multiple myeloma cells compared to our control, which is a delta scFv. The delta scFv has no targeting region, so the difference between these two shows a specific killing due to targeting of the CD229. And we can see that even at a 1-to-1 effector to target ratio, we see a very high amount of killing. So then the problem with this is the on-target off-tumor toxicity, or this low expression on healthy tissues. In our case, we have low expression on healthy T cells. And so we wanted to look at what that would actually do to the T cells if we treated with our CD229 CAR T cells. And so to do this, we used a mouse model where you inject human PBMCs into the mouse and then treat with our CAR T cells, and then later harvest the blood, and then we can use flow cytometry to analyze the surviving T cell number and also look at the staining of CD229 on the surface. And so as a control, we treated with both PBS or CD19 CAR T cells, and CD19 is not on healthy T cells. And as we would expect, we don't see any cell death for the T cells in these two groups, but we do see significantly more healthy T cell death with our CD229 CARs, and killing almost 50% of the T cells. This is very concerning. If you're killing 50% of a T cell population in a patient, you're very likely losing diversity, and this could affect the patient's ability to mount a T cell response against antigens that they've seen before, making them susceptible to diseases. And the other problem with this is CAR T cells are a living therapy that will hopefully be around in the patient for many months or years. And so if we're continually knocking out 50% of the T cells and knocking down that diversity, that could be a big issue for them. And so in addition to killing half the T cells, we're also selecting for a very specific population of T cells, or the CD229 low T cells. So we've done several experiments to show that these aren't being downregulated, but rather selected for here. And we recently published this paper showing that there's no significant difference of the CD229 high and low T cell phenotype, or the proliferation, but there is a significant difference in the reactivity of the CD4 positive T cells between the high and the low CD229. And so our main concerns then are both losing T cell diversity and also losing some of the CD4 reactivity of our T cells when we're taking out and selecting for those CD229 low T cells. So we need a way to select for just killing the multiple myeloma cells. And in order to do that, we want to take advantage of this much higher expression that we see of CD229 on the multiple myeloma cells compared to the much lower expression that we see on T cells. And you can see that the shift of this histogram on T cells is much less than the multiple myeloma shift. And this has been shown over many patient samples. And we're also just starting to quantify the actual number of CD229 copies on the cells and working on getting more patient samples for that. But even with this small subset, we do see that at least with 2, that we see a big increase in the number of CD229s on the cell. So our hypothesis then is that lowering the affinity of the CAR SCFV will actually retain the cytotoxicity against multiple myeloma cells while sparing T cells. And we hypothesize that this is due to the avidity of the cells still being high enough to kill multiple myeloma, but the avidity against the T cells not being enough to kill the healthy T cells. So for generating variants to test our hypothesis, we have to find lower affinity SCFVs that are still specific for CD229. So the way we chose to do that is to just take the CDR3 regions of both the heavy and the light chain of our SCFV and mutate single amino acid mutations where it's mutated into every other amino acid. And so we ended up with about 310 different variants of this with a single amino acid change in the CDR3 regions. And then we were lucky enough to have access to a high throughput SPR where we could then analyze all of these variants and get kinetic data. So we selected some of these variants based on their binding capabilities, all the way from 0.3 nanomolar to about 2 micromolar. And we tested their cytotoxicity against U266 cells at a pretty low effector target ratio of 0.5:1. And we can see here that we retained cytotoxicity against multiple myeloma cells with all of the different variants, regardless of their affinity, which was kind of surprising. And then since we retained cytotoxicity against our tumor cells, we then also wanted to see if we could improve the survival of our T cells. And we showed here that we didn't do that by changing the affinity of our SCFV. We see kind of a pretty similar amount of killing no matter which SCFV we're using. And so our hypothesis was that we could reduce the avidity by reducing the affinity of the SCFV. And the other way to do that, since this didn't work, is to reduce the number of CARs that are actually expressed on the cell surface. And so we replaced the strong EF1 alpha promoter for a weaker promoter in T cells called SFFV, and we looked at the toxicity there. And so again, we see here that with all of our different variants that we have, we do have cytotoxicity against the U266 cells at this low effector target ratio. And this is our original one. And so we have pretty standard toxicity. But that when we look now at the T cells, we don't see any killing of the T cells, which is exactly what we want to see. And so looking at these, we show that actually switching to promoter had a bigger effect on reducing the toxicity against the T cells than switching the affinity of the SCFV. Of course, we also see reduced cytotoxicity against our multiple myeloma cells here. However, this is a very low effector to target ratio, and so we want to go back and test this by just selecting a few of these and do more effector to target ratios and see if at a 1-to-1 we actually would retain the cytotoxicity against all the U266 cells, which seems likely, but then we're not sure what will happen to the T cells, but hopefully we still don't have enough avidity between our CAR and the T cells to confer toxicity. And that was a lot of data, so I just kind of took 2 of them, which is our original 2D3, and also one of our weaker affinity binders, and showed that the difference between the two promoters here against the multiple myeloma cell line for 2D3 is significant, and so is the survival of the T cells. And actually for this one, which looked the best with a binding affinity of about 1.8 micromolar, that we had a pretty significant difference in our T cell toxicity. And we still saw a good amount of toxicity against our U266 cells. So for my summary in future directions, it's really hard to find an ideal target for CAR T cell therapy. And one of those challenges is finding an antigen that's expressed on every tumor cell. In ovarian cancer, they don't have one of those yet. And so instead of finding one, we propose reactivating the CD95 pathway so that cells nearby in a solid tumor can also be targeted by the CAR T cells. And we did reactivate the pathway in SKOV3 cells. And now we want to see if the CAR T cells are actually able to kill those cells in close proximity. So that's what we're working on currently. And then for the CD229 project, we have this target that's on all the multiple myeloma cells, but unfortunately it's also on healthy T cells. So we're exploring the promoter effect more and working on quantifying the number of CARs per cell and looking at how that affects affinity modulation. And then we're wondering whether or not the CARs that express at a lower amount with this different promoter will still be enough to cure the multiple myeloma in our in vivo models that we've used previously, and if it will spare more of the T cells. So with that, I want to do my acknowledgements and thank Dr. Lim for being my PhD advisor and helping me with all these projects and giving me guidance, as well as the rest of the Lim lab. And of course, the Lutkins and Adenakovic lab for teaching me about all the CAR T cells and all the techniques that go along with those, my committee and my department, and then also, of course, the AFPE Fellowship and the Skaggs Fellowship from the LSAM Foundation. Thank you.
Thank you, Erica. I think we have time for maybe one question for Erica. Okay, if you think of one, put it in the chat. Let's finally move on to Dr. Franzini, who will introduce Julian.
It's a great pleasure to introduce Julian too, as a graduate student in my lab. He joined the University of Utah, coming from the University of California at Santa Barbara. In 2016, he joined my lab in 2017 and has since been working on developing new chemistry that can be used for either research in chemical biology or for drug delivery purposes. He has been very productive in the last few years. He's the first author on 4 publications, including some in the leading chemistry journals, and also 2 review articles. He has also been actively involved in both the department and the university, for example, at the admission committee of the BC program, just to give one of the many examples. He has also been a mentor of many undergraduate and high school students in this time here. And his productivity has been rewarded in addition to this Skaggs Fellowship, also by an AFPE Fellowship and a Graduate Fellowship from the University of Utah. He has also been the recipient of 2 prestigious travel awards, one for the Genelia Research Conference and one for a conference at the American Chemical Society. And he has also been received honorable mentions from the NSF Graduate Fellowship and from an NIH T32 in chemical biology. So without further ado, I'd like to give the word to Julian.
Okay, thank you, Raphael, for that kind introduction. And I will just share the screen. I don't really like presenting on Zoom. Raphael knows that. I really like it in person more, but you know, we'll see if there's any problems. My computer's been giving me some issues once in a while with Zoom, but hopefully it goes smoothly. So today's talk is titled Exploring a New Drug Delivery Platform and Enrichment Triggered Prodrug Actuation. So I'm going to focus more on a new drug delivery technique or platform that we are proposing or are hypothesized using reactive-based therapeutics. And I'll get into that a little bit more throughout the talk. And so as we all know, cancer continues to be a worldwide leading cause of death. This is the newest report from the World Cancer Report by the World Health Organization. And as actually Erica talked about in her talk, I'm literally focusing also on folate receptor alpha and ovarian cancer. Right, so ovarian cancer is proposed to about have 22,000 new diagnoses and about 14,000 deaths this year alone, estimated by the American Cancer Society. And there is that potential for targeted therapeutics, though, which can then lead to better therapeutics against ovarian cancer. And one target is what Erica mentioned earlier in her talk, is the folate receptor alpha. So usually chemotherapeutics remain as a first line of defense against many different cancers, not just ovarian cancer. But the problem is this dose-limiting toxicity, where these drugs are administered systemically, which causes severe side effects that limit the escalation of dose and the length in which we can actually administer such cytotoxic drugs against cancers. Now, the hopes are always against targeted therapeutics, where we can target the cancer cells and at the same time spare the healthy tissue. And one such target is folate receptor alpha. This one is overexpressed by many ovarian cancer cell lines, and against the normal tissue versus a malignant tissue, we see a stark contrast in their expression. And then usually it's correlated with higher disease state and higher histological grades, with higher folate receptor alpha expression. And so the way that this receptor works, though, is that this natural ligand is folic acid. So folic acid, as we all know, it's like a vitamin. And it is consumed in elevated quantities in proliferating cells because it is necessary in the biosynthesis of these nucleobases. And what's interesting and great about having this natural ligand of folic acid is that its binding affinity is extremely low to folate receptor alpha. It's low immunogenicity. It's easily modified to carry a drug. It itself is quite a small molecule, and it has low cost of production. So shown here is a figure of how cells take in folic acid. So most importantly, we can spare the healthy tissues that do not overexpress folate receptor alpha if we target it, because once the molecule binds to folate receptor alpha, it will internalize. So here's a short schematic. Here's folic acid is the ligand that binds folate receptor alpha. If you have a cleavable, some sort of linker to a drug, what happens is it will bind folate receptor alpha, and it creates this encapsulation or endosomal invagination, which brings it into an early endosome. There's an endosomal acidification step that then causes the release through some mechanism, depending on how you design your linker, which will then allow for the release of the drug and allow the free small molecule drug to then undergo its mechanism of action and induce cell death. So that is a basic principle of folic acid uptake in cells. And this has been targeted multiple times by many different groups using a variety of drug techniques. So here we have a mustard type drug or a platinum cross-linker or Taxol. So a variety of different prodrugs have been pursued to target using folate receptor alpha, right? So all of these drugs in common show here is folic acid as the targeting ligand. But these initial studies actually taught us quite a lot. So the what's most important though, so from 1 and 2, these do not have any sort of cleavable linker. And they actually showed that the mustard and the platinum, right, these are the DNA cross-linker type of drugs. They actually have less toxicity than the parent drug alone. So it's necessary to release the drug from the folic acid. And down here we have Taxol, and this one is an esterase or an ester cleavable bond here. And it's been shown that this one actually also does not have great potency compared to Taxol itself, which then tells us that it requires a linker with a bond that can be easily cleaved in that endosomal liquid or milieu. And then we come across to vintafolide, which is actually, it's a targeted therapeutic developed by Endocyte and Merck. And what you see here is, right, folic acid is here as a targeting group. There's a peptide spacer for hydrophilic and solubility reasons. And linked through here is this disulfide bond circled here in red to a vinblastine derivative as the toxic drug. Now, the way this thing works is when there's high amounts of, let's say, glutathione or thiols, endogenous thiols, it allows the cleavage of the disulfide bond and release of this toxic payload. Now, in phase 2 clinical trials, in combination with docetaxel, which I guess comes into a little bit play with what Erica was saying. A lot of these targeted drugs come with combinatorial therapy to target drugs that don't express high levels of your targeting receptor of choice. But in phase 2, it showed very great extension of survival over docetaxel alone. However, when it came down to a phase 3 clinical trial, they didn't meet the specifications that they were hoping for, and this was a proceed trial, it's what they call it, and terminated the trial. And so from this, though, many people in the drug delivery field have speculated that the disulfide bond, disulfide release, is not the most promising drug release linker due to the fact that endogenous thiols like cysteine or glutathione in circulation can cause the cleavage of this disulfide bond and release the drug prematurely. And so that is where we come in, and we think that instead of using endogenous factors, if we look at an exogenous trigger performing the bond cleavage reaction, we can get much better control over when and where the drug release occurs. Now, the chemistry required to doing so, is often termed bioorthogonal chemistry. It's chemistry that can be performed under physiological conditions, right, without engaging endogenous functionalities or perturbing natural functions. It's inert to the proteins and et cetera around in the body. Now, the example shown here is for a pre-targeted approach where instead of, for let's say imaging purposes, especially shown here, it's radio-labeled antibody. It takes time for the antibody to circulate and then finally leave the body. So as you can tell here, it takes 48 hours until you get a good signal. Now, the pre-targeted approach is if we start with an antibody with a reactive chemical group, allow it to go to the tumor, and then wait some period of time for it to clear the body. And then we add in an exogenous reactive group, which here is tetrazine to a radioligand. The ligation reaction between the two will occur and give you a very great signal over background. Now, so that was one example of some sort of therapeutics that bioorthogonal chemistry has shown promise in. But as I mentioned, right, that one's a ligation reaction. What we as a group is interested in is if we can transform these reactions into a dissociative one. So instead of forming that stable ligation product, as shown in the previous slide and here, we form a labile ligation adduct that will further downstream release a caged molecule, for example, a drug. Now, this has shown preclinical success in using antibody-drug conjugates with a bioorthogonally cleavable linker instead of relying on the endogenous like protease-style linkers. So in a study done in 2018, they demonstrated that this antibody-drug conjugate that was linked through a exogenous tetrazine-responsive linker showed to lower the tumor volume considerably compared to an antibody with a protease cleavable linker that was used in the FDA-approved Kadcyla drug. And so what's more interesting and nice to look at is literally yesterday, this paper was published in the bioarchives, but also this news report came out that click chemistry will see its first use in humans. So these reaction-based therapeutics are going places. And the first trial is beginning here in the United States and in Australia. And you can, if you want to look into it, the clinical trials identifier is listed down here. And this is a prodrug activation technique where they use biopolymers and a release of a prodrug in the vicinity of a solid tumor. But I'm not going to go into too many details on this one. And instead, I just want to introduce isonitriles, which are the chemical group that our lab is very interested in as a bioorthogonal functionality. Now, there's many different chemistries out there that can do this sort of bond dissociative type chemistry. I'm not going to go today into the pros and cons of them, but our lab focuses on the isonitriles. And we and others have shown it to be stable in biological fluids and show moderate to good metabolic stability. It's non-toxic, it's chemoselective with tetrazines, so they don't react with the endogenous functionalities. It's synthetically accessible, and especially it's structurally compact, so we think that it would minimally perturb the pharmacokinetics of the parent drug if we make them into prodrugs. And so based on the isonitrile, we have in our lab designed and published a few different drug release chemistries. One is called 3-isocyanopropyl, which is this group and with this gray ball representing a toxic payload, for example. In reaction with tetrazine, it undergoes a series of steps to result in a 3-deoxypropyl group, which is known to undergo a spontaneous beta elimination at physiological pH to give you your free drug. Alternatively, we put the drug onto the tetrazine side and created what we call tetrazyl methyl groups, which react with isonitriles, and again undergo a variety of steps to form this 4-aminopyrazole molecule, which can then spontaneously undergo a 1,4 elimination step to release again your free payload. And in collaboration with Randall Peterson's group here, and especially thank you to his postdoc, Dr. Saba Parvez, we were able to demonstrate that our chemistries, both of them, the tetrazyl methyl and the isocyanopropyl chemistry, are able to be used in a living system by the dual release of these fluorophores in live embryo, zebrafish embryo. Now, taking this chemistry, though, we're interested then to apply it back to disease states like ovarian cancer. And how we're going to do that is based on very similar prospect is shown here. This is enrichment triggered release. So a paper published in 2018 showed that when they were targeting mitochondria using TPP, triphenyl phosphonium ions, this lipophilic cationic probe to target the mitochondria, they were able to enrich their bioorthogonal 2 molecules into the mitochondria and reduce what would have been a reaction that takes with a half-life about 100 hours down to approximately 2 hours. Now, again, this is targeting the mitochondria. Like all cells have mitochondria, so it's not a viable target for us to enrich our probes for drug release purposes. But we hypothesized, right, that if we target folate receptor alpha, as demonstrated here, the internalization of our reactive probes and trigger molecules into these early endosomes will give it that proximity enhanced reaction rate, triggering our dissociative bioorthogonal reaction and allowing for drug release to occur in the endosome. And what's important to note also, as shown here in this figure that I wrote, for example, folate receptor alpha PSMA. So this concept, though, is what we believe is extendable to other surface expressing receptors that upon binding will internalize into these early endosomal structures. So PSMA is prostate-specific membrane antigen, and it's widely expressed on a variety of prostate cancers as well. So for example, there's also other targets that we may go after in the future. But again, we are looking at the concept of internalization, enrichment triggered by the internalization of these targeted receptors. So I walk you through a little math. Based on the literature, we find that on average, you can get about 10 picomoles of folate receptor alpha would be internalized per million cancer cells, which means this 1 × 10⁻⁻18 moles of folate receptor alpha internally per single cell. It's also been measured that these endosomal, total endosomal compartments will comprise of approximately 6.8 femtoliters of liquid, right, per cell. And if we just divide the moles by the volume, we see that we should be able to get an enrichment of concentration of approximately 0.15 millimolar within these early endosomal compartments for our reactions to occur. Now, the reaction we're using, the tetrazyl methyl chemistry, we've measured a bimolecular rate of about 2 per mole per second. So if we started with a concentration of 10 micromolar, the half-life would be about 10 hours in circulation. So it's very slow. And so the reaction would not occur before it leaves the body, for example. However, if our hypothesis is correct, binding of these probes and giving it the concentration of 0.15 upon internalization, this reaction would then only have a half-life of about half an hour within the cells. So to test that, we made a variety of probes. So this one we called tetrazyl methyl fluorescein folic acid. So this is the folic acid part, a long PEG chain connected to a fluorescein dye and our tetrazyl methyl. Now, when this fluorescein fluorescent molecule is caged on both sides, the fluorescence is turned off. Upon release of the tetrazyl methyl, we get HO or hydroxy fluorescein folic acid, which then turns on a screen fluorescence. And as the trigger, we have folic acid isonitrile. So this is folic acid here, again, connected to an isonitrile. So the reaction between this molecule, folic acid isonitrile, and the tetrazyl methyl should lead to this hydroxy, which is also what we used as our positive control. And so, yeah, we took the positive control and we were just looking at the concentration dependent to see what we can get at a minimum. And the reason why we looked at that is because these receptors, or the KD, is so low. We don't want to completely saturate. We just want to use the minimal amount to get a reliable signal. And so we stuck about 2 micromolar. And from there, we started looking at the time dependence of the reaction. And with 2 micromolars of the tetrazoyl methylfluorescein folic acid with 10 micromolar of the folic acid isonitrile, at 4 hours, the signal is barely noticeable. And at 12 hours, we start seeing somewhat what we believe is the reaction occurring. But again, it's very non-significant at that point. But the longer we waited, all right, so this is at 12 hours, and this is the positive control incubated for 12 hours. And as you can see, while it is shown significance over the negative control, which is incubation with tetrazoyl methylfluorescein probe alone, it is far off from the positive control. However, longer incubation periods up to 24 hours, we start seeing a signal that is comparison to the positive control that's been incubated for also 24 hours. So while our math has shown us that theoretically we should have a faster release kinetics, we are still seeing the release occurring due to endosomal internalization of our probes. Now, the next steps we'll need to look at, though, is to block the receptor sites by saturating folic acid to make sure that this is folic acid dependent release. But it is very promising for us to see, although the kinetics are a little slower, we found that these results to be very promising, that the reaction is occurring within the endosomal due to the appearance of punctant fluorescent signals within the cells and the growth of the fluorescent signal over time. And so a few other things that we should look into, though, is the log phase of the cell growth versus incubation during stationary phase, because it's shown in literature that the expression level and internalization rate changes with the phase of which the cell is growing. There's also other cell lines that express folate receptor alpha to a higher degree that we may look into. But most importantly, I believe we should look into the different linker lengths, as I highlighted here, because there is that step that needs to either acidify to wash it off the receptor, or if they are still bound to the receptor, we are unsure at this point whether or not the isonitrile can reach the tetrazoyl methyl for the reaction to occur. So looking into alternative linker lengths will guide us a little bit as we probably will lengthen the linker and see if we see any significance in kinetics difference there. Ultimately, we would like to transform this system into drug release, right, as I talked about in the very beginning. So how that would work is we would still use the folic acid, and instead of releasing just a fluorophore as a test molecule, we would redesign it to be a tetrazoyl methyl linked to a drug molecule here as shown here, and upon reaction with the isonitrile should release the drug within the cell. And that is where we hope to go. And again, instead of just folic acid, there are also other ligands that we find are very promising, like PSMA or as well as CA9 for future directions. And with that, I'd like to thank my lab, PI Dr. Rafael Franzini, some of my lab members, Titosh and Braden, the Peterson lab, especially his postdoc, Dr. Saba Parvis, for his help in our initial chemistry development studies, other labs in our department that has helped me with equipment, etc., and the remaining members of my thesis committee, Dr. Darrell Davis and Dr. Mingmen Chen. I'd like to thank the Drug Discovery Corps for high throughput imaging, medicinal chemistry front office, an AFP fellowship, and especially the Skaggs Graduate Research Fellowship and the ALSAM Foundation. And with that, I'd like to take any questions. Thank you.
Julian, great work, if I may ask a question. So, you know, the endosomal uptake and basically that endocytosis process, we're talking about minutes, right? Whereas you saw after 24 hours, you get that increase in fluorescence. So what is the reason for that?
Right, so that is, when I was talking about my future direction slide, that could be, actually, let me go back to the, I didn't draw this figure, but is So yes, the internalization step into that endosomal compartment is very fast. And based on the literature values that I have been able to find for concentration and volume, the reaction that I'm hypothesizing is supposed to occur within that half an hour to an hour range. But as you said, I mean, I've not seen a significant, very high significant signal until 24 hours. And that's why I also was talking about for linker lengths, because if the receptor is still bound on the surface, I'm not sure if they can actually reach each other. But here, shown on this slide, there is this endosomal acidification step at proton pumps, and that changes the protein conformation of folate receptor alpha and washes the folic acid off the receptor. And at that point, then they can react with each other. But that step is not as instantaneous as the endosomal formation. And so the recycling rate is about 8 hours. So at that point, I'm actually not entirely sure at what point in that 8 hours is the acidification step, but without it washing off the walls, if it's still bound, I'm not sure if the reaction is actually occurring until they can reach each other. That's why I'm going to explore different linker lengths as well. And I think there's a lot of other reasons too. I mean, there's some things that are out of our control completely, like the stochastic binding of our probes, right? I'm using about a 1:5 ratio. And if, for example, in one endosome, there is a 1:100 ratio, just random binding, and then the reaction rates, again, is completely skewed. So there are a lot of different aspects of this project that still need to be developed and looked into. But we found that the results was promising that the reaction is occurring, yes, although at a slower kinetics than the literature values that we hypothesized.
Great, thanks. And I have a follow-up question. So as you know, there are some other pH-sensitive bonds that people have used, for example, attached to polymeric systems or even small molecules, such as hydrosome bonds. So what are some of the distinct advantages of your bond versus some of these other pH-sensitive bonds for intracellular delivery?
Oh, well, so for intracellular delivery, like we were saying, it's not a huge difference in that sense. But for example, when you're talking about acid-sensitive bonds, it's not just within the tumor that is acid-sensitive, right? If we just start with an acid-sensitive bond, it's well known that in tumor microenvironments in general is an acidified media. So we can get drug release earlier than expected. But instead, if we use an external trigger like our system, right, there is no drug release until the reaction occurs. So we're not dependent on any other environmental factors other than our reaction. So it gives us much higher control over maybe release of the drug elsewhere than what we actually want.
Thanks. Great job.
Thank you.
Julian, this is Sava.
Yeah.
Yeah, great job. One question: how are you treating the cells? Are you doing co-treatment of your two components, or are you doing one and then washing away and then treating the other, or do you see release in situ? As you treat.
Right. So I didn't show it in this presentation, but I'll stay on this slide, right? The KD for natural folate receptor is like nanomolar range. So that binding is very, very tight. And the way people wash it off, though, is with an acidic wash. So when I tried it initially, I used 2 micromolar versus 2 micromolar of our caged probe and 10 micromolar of our trigger. When I did them in sequential, like tandem, like put in one probe and then the next, or vice versa, either one, sequential order, I never saw any turn on actually. And what we hypothesized from that is that we're saturating the entire receptor right away. And instead, in order to see some signal, when we were seeing is a pre-mix, if we pre-mix them and then add it to the cells, we are starting to see some signal appearance. So no, I'm not washing in between steps. I pre-mix and load onto the cells. And then we have that stochastic binding.
So Julian, that experiment where you did it sequentially seems like an important experiment, right? Because that shows that if you do it sequentially, you basically saturate the receptors and there's no competition. I mean, so there you have competition essentially in that the other component doesn't ever bind the receptor and get internalized. And so I'm just trying to think about why this isn't working better, right? It just seems that there's a fundamental problem with competition for the receptors from the two different folic acid moieties. And one could imagine if there was if there was a preference for one over the other, then the tighter binding one would saturate the receptor and you never get the second one in there and you wouldn't get the reaction.
Right. There should be a preference. I've actually never measured our probe's KD, but based on literature, the molecules do fluctuate very slightly in preference. But if it is a mixed group or a mixed solution, we should expect to see some sort of distribution between the two probes, though, that gets internalized. So I don't believe if I do it in tandem, I think that it would be saturated, and that's what I believe I am seeing. But if it's a pre-mixed solution already, like if we go towards let's say in vitro IV injection, it would be a mixed of the two already, but it would be at such a low concentration that the reaction does not occur while in circulation until it's internalized.
Yeah, I understand that, but if you had even a tenfold difference in binding affinity, then you're going to totally have a preference for that type of drug to get internalized.
Right, but based on the literature, again, I haven't tested the KD of my probes per se, but based on the literature, when you put different drugs at either the gamma or the alpha site, it is I have never seen in the literature a tenfold difference in KD. It's always within 1 nanomolar up to about 5 nanomolar differences, very slight differences in their KD values.
Yeah, I'm just thinking that there doesn't seem to be, at least there's not an obvious way to evaluate the internalization of a particular compound. The only way you really can tell is once they react. There's no sort of independent way of saying, oh yeah, this one gets into the endosome better than some other construct.
Oh, I mean, the reactive one's a little bit different, but if you put different like fluorophores that are already on, for example, instead of caging them, you can track the probes themselves if you really were interested in that. And also a lot of people that work with this, they use tritiated folic acid. So it is able to track if you really wanted to track the two different probes. Yeah.
Okay, thanks.
We are very rapidly approaching the top of the hour, so let me express hearty congratulations to Danielle, to Snigdha, Erica, and Julian, and thank them for their wonderful presentations. Thanks all of you for participating and all the questions. I believe that Ryan Boyack has some closing thoughts for us.
Yes, I'm gonna share my screen. I appreciate I appreciate everyone that's prepared for this today. The impact of this gift has been great. This is the 4th year that we've had the Skaggs Scholars receive their awards from the endowment, and there's many more years to come. And just the impact of this gift on research is incredible. So thank you again to the Skaggs family and Ron Kutchell for all your help with this gift. And it truly has been something that has made a huge difference at the College of Pharmacy. So in closing, just give me a minute. We're gonna give out everyone, I think we got everyone's name on this. We're gonna give out a couple protective face masks, and we'll spin the wheel. We've got 4 of them to give out, and I'll make sure that I get it to your, oh, there we go, Sean Owen. Congratulations, Sean. So let's see. And then Ting. We got 2 more to give. Ooh, and our last one, Katie.
Don't worry, everybody. This is not how we select the Skaggs Scholars.
Yes. Anyway, thank you so much for attending. Have a wonderful day, everyone.